I do teach, train, and conduct research in, in embryonic stem cell biology. Thank you, I'm flattered. First of all, I have the honor to moderate today's panel. And I'll open with a brief basic introduction to what stem cells are about. And then we'll be joined by 4 of my distinguished scientists in the field of stem cell biology, who will be given a series of 4 lectures. So please hold your questions, note them, and make sure you remember them. We'll be all sitting at the end of the presentations, and we'll be very happy to answer all the questions that you have for us. So we'll start a little bit with the stem cells. And this is, it's not moving for a reason. Thank you. Technologies sometimes give up on us on a weekend. It's okay. Stem cells. Gil had the introduction given to you this morning, and I will, I'm an educator, and I believe that the more we repeat, the better. So I think that the more you hear about it, the more you'll get more details, and said in different ways, it would be complementary. Stem cells are unspecialized cells. I always call them the students of the body. They are a sort of undeclared. They still don't know what they're going to be. So they spend their time, most of the time, dividing because they can. And from time to time, they can decide to differentiate, specialize, and give birth to one of those more than 250 type of specialized cells in the body. Now, those stem cells, we have them with us. The ultimate stem cell is basically the fertilized egg. As you know, that egg can divide and give birth to the whole body. So it's pluripotent. It has a lot of potential for that. As we grow and as our body is formed, most of the cells have the time to differentiate. And once they differentiate and they specialize, they tend to have only a certain number of divisions they can go through before they stop. That number is about 50. But we still keep some cells in some niches around our body, and those cells can keep dividing for a long time. And that's a feature they share together with the cancer cells. Those are the 2 types of cells that don't stop at the red light. They keep dividing, and one of the good things about stem cells is by understanding how they work, we can also understand how cancer cells develop, and this is in addition to the regenerative features of these cells. So in our body, their role is mostly to renew the body and replenish other cells that are either worn up, dead, or wounded. As you know, there are 2 types of stem cells. The adult stem cells and the embryonic stem cells. So I'll start by defining a little bit what adult stem cells are, and we'll move on to the embryonic stem cells. I'll try again. Oh, it's already done. Stem cells, the source are, as I told you, they are in our body. We have areas of our body that have a lot of stem cells in them, mostly the skin, the digestive system, but also the brain has some areas that have still some adult stem cells, but the most known are the stem cells of the bone marrow. They are the ones that have been used for a long time, and they are the ones that are the most known among general public. These cells do have one a little disadvantage, is that they are limited. They are not like the embryonic stem cells. They are only pluripotent. Meaning that they have less potential to differentiate into a wide array of tissues. And most of the time, they can only give birth to the same type of cells as the organ from which they come. Next, please. Another source of stem cells where we can actually find stem cells are the umbilical cord and placenta. Now, any couple or person who goes to a clinic to give birth are given the option of whether they want to save the cord blood stem cells for their children. What you need to know about those is that 90% of those cells can only give birth to blood cells, because that's what the origin of those cells are. However, there are 10% that can be plastic, and still research is going on to find out what kind of flexibility the 10% have. However, in terms of number, the umbilical cord is not very long, and therefore the number of cells that we tend to extract for them is not enough for an adult. So there is a problem with numbers, and there's a problem with the fact that we cannot multiply them, we cannot grow them. More research have shown that other sources can be found. One of them is the amniotic liquid. That's the little liquid that surrounds the baby in pregnant women. Have been shown that they contain also some adult stem cells. And more bizarrely, recently, have been shown that menstrual blood have stem cells. Not to the level where your lady can start saving your tampons, but it's a very good option. Treatment of, an example of treatment with stem cells would be something that's used for 50 years so far. For anybody who has to deal with leukemia knows that leukemia can be treated by a bone marrow transplant as long as you can find someone who match. So the way it works is the cells of the bone marrow, which are mostly hematopoietic cells, those are the progenitors of cells that will give birth to blood cells, can be collected from a donor, cultured, and then transplanted directly into a patient. Those cells tend to know where to go. They tend to go to where what used to be the bone marrow of the patient, recolonize it, and start making cells for that patient. So as long as there is a matching, as I said, there is no risk of rejection. But as you know, matching is very rare, and it's almost 1 in a million chances of finding someone who matches you. Next, please. So advantages of stem cells, the mature ones at least, the thing we know is that they are adults. So they know what they want to be, and they tend to be almost inflexible. So they are partially specialized. We don't need to tweak too much with them. Culturing them, we know where they would go and what kind of cell tissues they will make. So that makes it easier in terms of what kind of chemical manipulation you have to do with them to be able to direct them. Some are easy to find. The one in the bone marrow are easy to get. However, the others are a little bit difficult because we cannot recognize them, and I don't imagine someone allowing the doctor to go to their brain to try and collect the 3 stem cells that we have there. So access to some of those is a little difficult. Advantage another one is that same type, if someone uses their own stem cells, for example, pre-chemotherapy treatment, someone gives the stem cells and we get them transplanted after the chemo, will be rejection-free. However, those cells, and unfortunately, they have also disadvantages. Next. One of the disadvantages of adult stem cells is that they have a limited longevity in cell culture. I told you about that number 50. When you have an adult stem cells, you don't know in what stage of division it is. It could be in 48 and has only 2 divisions to go for, or it can be in stage 1 and you can grow it for a while. But there is always, in general, a problem with growing them and proliferating them in the lab. I told you about the problem in isolating them and extracting them because of limited access. They are limited in their flexibility, so we cannot make a lot of cells out of them, although a lot of labs are involved in trying and find out how to manipulate those cells to make them do more than they think they can. They are very rare in terms if you compare them to the number of cells we have in our body, and they tend to be limited in terms of numbers as we age. We lose a lot of the stem cells as we go through the age. Another factor that affects them is the lifestyle. As you know, they live with you, and therefore they're going to be subject to everything that you do to your body, from smoking to drinking to the pollution of 101 if you drive every morning to a commute. That's why the option of embryonic stem cells came. It's because of the fact that adult stem cells cannot offer everything we would like them to offer. Next. So embryonic stem cells, as you know, and as their name indicate, comes from an embryo. That's a fertilized egg that has developed for about 4 to 5 days. After it develops, it makes what we call the blastocyst. You can see it here how it looks like. A blastocyst is made of an inner cell mass. That's the one we collect the stem cells from. And then there is a trophoblast. This is the one that allows the embryo to implant. So stem cells themselves cannot make humans because they cannot implant. Stem cells are made to be manipulated to make other kind of cells for patching purposes. So the myth of making little Hitler soldiers from a little embryo collected from a hospital is a little bit far away from anything other than a myth. The cells are collected in a lab, and they can be incubated in incubators, and they can be maintained theoretically forever because they do not, as I told you, answer that 50 number of divisions factor that applies to adult stem cells. Next. There are several sources of embryonic stem cells. The one that was used to originally extract the stem cells is the in vitro fertilization. People who cannot conceive naturally can have that done outside their body, the test baby of the early 80s, 90s. The way it works is a donor, a mom in general, gives the egg. It doesn't have to be the mom. Sperm is given by the male, and then both of them, the fertilization is made in the lab. Now, when fertilization, when a couple goes to get embryos, they tend to use more than one. The procedure is difficult, and therefore it is recommended that you fertilize more than one embryo. However, you're not going to transplant more than 1 or 2. So there's always some leftover of embryos from any IVF procedure. As a matter of fact, there are 400,000 embryos that are stored frozen in hospitals around the US. And ultimately, when the couple is not needing those embryos anymore, they will be discarded. So the first stem cells were isolated in 1998 at the University of Wisconsin, came from fertilized egg that were originating from IVF clinic. However, this procedure, because involves the destruction of an embryo, there are groups that are opposed to it. So scientists have been trying to a little bit override this ethical or controversial problem. Recently, advanced cell technology have shown that they might have a way of extracting stem cells from fertilized egg without destroying the embryo. That work is ongoing. We are still waiting to see if they can actually create stem cells from that procedure. Another one that's also more recent next is to try and make pluripotent cells. Those are cells that look like an embryonic stem cells, but they're not completely embryonic stem cells, by trying to reprogram adult specialized cells, such as the cell of the skin. This technique was developed recently at the University of Kyoto and the University of Wisconsin together in a separate studies, but they show that that's possible by introducing some growth factors in skin cells from an adult patient. You might be able to make embryonic-like cells. What you need to know about that is in order to introduce those factors, they had to use viruses, and we still don't control very well what a virus would do in your body. So this is a long way to go before it can be used for any transplantation. So far, the most promising technique in stem cell biology, next, is what we call somatic cell nuclear transfer. This is a scientist's word to replace what we used to call cloning. There are two types of cloning. One is reproductive cloning. It's the aim of making another body that's banned and nobody wants to do it anymore, anyways, I mean. And then there is the therapeutic cloning. That's the cloning for the purpose of making cells. And this is what that is. This involves an egg from which the nucleus can be removed. The eggs become a surrogate egg, and we can replace the nucleus by a nucleus from a patient's cells, for example. So what you have in the end is a little embryo that does not involve sex, and that embryo can make stem cells that are custom-made for the patient who gave this nucleus. Next, so we can imagine how this kind of technique will be put to use. So far, it has been used in several species, recently even in primates, and there is still work. There are some indication that it might work in human, and I'm guessing that Dr. Reijo Pera will have some more to tell us about this, as her lab is one of the world experts in this technique. So let's imagine someone has diabetes, but it doesn't have to be only diabetes. That can be Parkinson's and Alzheimer's and spinal cord injuries and stroke. So the embryo will be made from, the stem cells will be made from the blastocyst of an embryo that was made, custom-made through this, the SCNT technique. Those cells can be manipulated in the lab to become one of those specialized cells. Here we have the example of a pancreatic cells, and we can imagine that those could be injected in a patient and go replace the endogenous pancreatic cells of the patient that probably were lost or diseased, which is the origin of type 1 diabetes. Next. So you get a little bit the point from the embryonic stem cells. They have several characteristics that cannot be provided by the adult stem cells. They are first easy to grow in culture, can be maintained for a long time. They are very flexible. If we know how to coax them, we might be able to make a lot of cells. And they have that potential of being clinically relevant in terms of regenerative medicine for a wide array of diseases. Next. So this is a chart showing you what so far we can do with embryonic stem cells. They can be grown in a dish, and as long as we know what the protocol is to make them, to direct them towards more specialized cells, we hopefully one day will be able to make a homogeneous culture of specialized cells that can be used for regenerative therapy. But so far we know, for example, how to make cardiomyocytes, we know how to make nerve cells, blood cells, pancreatic cells, and even hepatocytes. Those are the cells of the liver that could be replaced for someday override the need for transplantations. Next. Let me give you, just to finish, some examples of what we do here at Dominican. This is how mouse embryonic stem cells, which we culture here in my lab at Dominican, every little circle of this is a little embryonic cell. They can grow, and this is a dish in which they are growing. You can see that there is still some room, but as you let them go, they can grow over and over. So you can actually collect them as you go, grow them and collect them, and have an infinite amount of cells. For example, this cell line that I'm using was made in 1990s, so it has been used for a while, and those cells are still growing since the mouse mom gave these cells and died maybe sometimes in 1988. Next. So at Dominican, we culture them. We know how to turn them into nerve cells, which you see here. And this is all done by undergraduate students that are trained to do this kind of technique. My project in the lab is to use these stem cells to understand how a certain type of nerve cells do develop. They're called the neuroendocrine cells. Those are the hormone-secreting cells in our body, and they control from stress to metabolism to growth to reproduction, some of the most vital cell functions in our body. The problem is we don't know how they grow, we don't know how they specialize, and we don't know why they become what they are. So that's one thing you can actually do with stem cells, is use them for biology to understand the normal development that's happening in the embryo, which would be otherwise very difficult to study given the invasive type of techniques you would need to use. Next. So these are the cells I showed you that we can make neurons, and we can even find that some subsets of population of those neurons do express markers of neuroendocrine cells. So this is our model that we use here to both train the students and use these models through some gene silencing to find out how they develop. Next. Other labs have actually focused on other elements of what you can make with stem cells. This is human stem cells grown at the University of Wisconsin by the laboratory of James Thompson. And they have been coaxed to become cardiomyocytes. Those are the cells of the heart. So you can imagine an undergraduate or a graduate student working in a lab. It's 10 p.m. or 11 p.m., nobody's around, and you look at your dish to see how your cells are doing, and what you find is something like this. So these are beating heart cells in a dish. So we know how to make them. The only problem is when you put them in the body, they tend to go to the heart and replace cells that are actually dying. The only problem they do is they are immature. And as you know, the baby's heart beats at about 120 per minute. An adult is about 70. So when they go to the heart, they stick to the heart, but they start beating on their own. It's just like putting the symphony orchestra with Metallica and asking them to play their own tunes together. But this summarizes a little bit the promise of stem cells, but also the pitfalls with stem cells. As you can see, this is the part that actually did differentiate into heart cells. And what about the rest that did not differentiate? If this is transplanted as it is, we don't know what kind of cells we're putting in the body. And as you know, a stem cell that's not differentiated can be the origin of a tumor. So there is a lot of work to be done in how to coax these cells and how to make them become homogeneous, meaning we have to make sure that what we're transplanting into the patient is 100% homogeneous cell that have specialized in what we want to. Just before I finish, I want just to do a little hi to my students who've been helping me in the lab through this work. David, next. They'll come. But thank you very much for, for your attention.